Everybody can hear me. I'm really excited to present this research in conjunction with Cascadia. I did an internship with them right before I came to Evergreen, which was actually about 4 years ago at this point. So I'm very excited to present this research, get this done, and I hope you guys really enjoy it. I'm going to talk about rough-toothed dolphins, scientific name Steno bredanensis, and their pigmentation patterns and how pigmentation indicates community associations. So you're going to see some dolphins today, which is exciting. Starting off, I'm going to talk about my study species, which is the rough-toothed dolphin. They are pictured to the right, at least it's the right on my screen. I hope it's on yours as well. They have a ton of pigmentation, as you can probably see from this photo already, which I will talk about a ton more. And pigmentation is any of those kind of dark markings. The ones I look at are mostly in the cape and the dorsal fin area, two areas I will define more. Rough-toothed dolphins occur worldwide in tropical and subtropical waters. They're generally a pelagic species, so an open ocean species, and there's not a ton of research that's been done on them because of their open ocean status. They are very hard to find. The ocean is very big. They tend to travel in large groups. However, there are some insular populations, which are island-based populations, and those are the ones that I looked at in this study. And they're currently worldwide managed as one stock. Obviously, there's not a fishery for rough-toothed dolphins, but when looking at kind of the threats that rough-toothed dolphins face, they're managed as one stock. However, there has been some research into whether at least the Atlantic and Pacific populations should be split. So it's kind of ongoing, as it is with a lot of cetaceans, lots of whales, kind of trying to figure out what subspecies exist, what species should be separate. That kind of stuff is very ongoing. Like I said, there's limited research into them in Hawaii and worldwide, particularly their pigmentation. So Hawaiian Islands, most of us are pretty familiar with them. That's where a lot of research effort has occurred because there are a lot of odontocetes, lots of tooth whales that exist in that area. Also, if anyone puts a question in the chat, please let me know. Sorry, I just see it popping up and I just want to make sure. So I looked at 3 populations in Kauai, Niihau, that top left arrow. So those 2 island areas, Oahu and the island of Hawaii. And that's where most of the research effort that Cascadia has done in the last 10 years approximately at this point has been occurring, mostly over like working with false killer whales and whales that are endangered. However, they're taking pictures of everything, and I'll talk more about kind of photo ID and those things in a second. Rough-toothed dolphins exhibit philopatry and high site fidelity, which are 2 fancy words that mean they basically hang out where they were born, in Hawaii, that is specifically. So they have a strong island association. They're not kind of going back and forth from Oahu to Hawaii. And that's really important for the stuff that I looked at, the pigmentation. And like I said, there are different populations for each island, and that's Hawaii. So pigmentation. This is a good example of one of the pictures from the catalog that I used, which shows the pigmentation really well. Rough-toothed dolphins have unique pigmentation to the individual, so every single dolphin has like a fingerprint basically, and it does not change over time, which is true in other whales. For instance, spotted dolphins, they add spots over time. That's not the case with rough-toothed dolphins, so it makes it very useful for identifying them and saying this individual we saw this year is the same as this one we saw yesterday, which I'll talk about a little more. And there's really no research on pigmentation and how it changes, how it changes populations and things like that. So photo identification is kind of the basis of where my data came from and why it's important. It's a super commonly used research mechanism. And the picture shown is actually one of Cascadia's research boats, which I was lucky enough to get to go out on once in Hawaii. And there's someone there taking a picture. And as you can see, you find a big group of whales, and whales or dolphins, and you start taking as many pictures as you can. So it involves a lot of work in terms of isolating individual photos, because all the photos that I went through are individuals. And so you compare that with an existing catalog to get a match. And there can be a lot of uses and applications for that, not only in like rough-toothed dolphins, but in all like whales and dolphins, cetaceans. It's super commonly used, both for like biological studies, to see how long animals are living, if they're staying together, like in groups called dyads. Just a ton of applications. And the best kind of thing we have, because it's used boat-based, shore-based, or aerial observations, it's kind of one of the best things we can do with an ocean so big. This is another really great photo from the catalog that Cascadia provided me with, that shows the two study areas that I focused on the most: the dorsal fin, which I believe most of us are probably pretty familiar with, the one that's a little more straightforward than the cape area, which is defined as kind of the flank area. And when I was doing my research, kind of drew those lines from the inflection points of the dorsal fin, so where the dorsal fin connects with the body, kind of to create a sort of trapezoid, so that I could make an accurate estimation of the percent of that dark gray cover. So what I was looking at is the dark gray on top of the light gray. There's a lot of gray going on for sure. So kind of looking at what that is, because that's the unique part of it. And the bottom line that I use is called kind of the midline, where that dark gray goes into a light gray. It's kind of difficult, especially with the cookie cutter shark bites that are down there, but I'll have more pictures that should make it a little clearer, hopefully. So my research question is there a relationship between pigmentation pattern and island association in Hawaiian rough-toothed dolphins? So I'm looking at, is the percent cover and the island association connected in any way, or is it just random? So for my methodology, like I mentioned, I used the research catalog that at the time of my research had 2,378 individuals present in it. And that number was growing every day. I'm sure it's bigger now. And I coded 1,311 individual photos. The ones I didn't use of that, approximately 1,000, were not of good enough quality for me to be able to say for sure, like, this is the amount of percent cover. And so I estimated percent pigmentation cover in each region, those two regions that I just showed, the cape and the dorsal fin. And I binned those in 25% intervals because I was using a kind of an estimate of the percent of that dark gray cover. I used a chi-square test to determine the significance of my data at an alpha of 0.1. And to the right is the number of individuals from each island. So Hawaii and Kauai Nihau had a pretty large number, Oahu less, and that's reflective of the research effort that's occurred in that area. There's been a ton of research effort in Hawaii and Kauai Nihau, just less in Oahu as of this time. So I'm going to go through two different fins and kind of show you what I coded them when I went through. So this one is actually a really great photo because it's straight onto the camera. There's no angle. With all of these photos, even the quality ones, there can be angle issues, reflection, any sort of thing that make it a little difficult to code it. However, this one's got a pretty good trapezoid because you can see the entire midline. You can also see kind of that horizontal inflection point where the dorsal fin becomes the cape and vice versa. So with the fin, this was in the 25 to 50% bin, and with the cape 75 to 100% because there's a lot of cover going on. And as you can see, like, that pigmentation doesn't obviously stop where I stopped looking at it. There's more going down the length of the body, but almost all of the pictures I had were only of this area or even less. So I'll talk about that a little more in terms of more research that with these guys. This one has a little bit of an angle to kind of show you the variety of pictures I looked at. So for the fin, this is in the 0 to 25% bin. There's really not a lot there. There's some color discrepancies, but nothing that's that really dark, grayish, black almost, that I was looking for. And the cape for this one is 25 to 50. The like trapezoid triangle or box thing is at a little bit of an angle. But again, with the estimation, I was doing kind of the best I could with what I was seeing. So these are all of the kind of codes or percent cover that I initially looked at. The ones inside of the box are the ones that I did an analysis of. The ones that were whole in pigmentation, wispy, pigmentation below midline. I just didn't have enough data points to feel comfortable making an analysis of that, but they were all patterns that I'd seen through my time doing photo ID on these individuals. So I would be interested if they were actually something that could be examined further in the future. So moving into my results, like I said, I looked at cape cover and dorsal fin pigmentation. So I'm going to start with cape cover pigmentation. So like I said, I did a count of these individuals, and that's shown here in a mosaic plot. And it shows the proportion of the individuals inside of each kind of column. So each column is all of the individuals within the 25 to 50% range, and then which ones were in which island. So the interesting thing about the cape pigmentation is it shows this kind of stair-step pattern between Hawaii and Kauai Nihau. So there are a lot of individuals, a high proportion of individuals in Hawaii with not a lot of cape pigmentation, so not a lot of that dark gray, versus in Kauai Nihau, there are a lot of individuals with that dark gray on the cape, so with a higher percentage of that. And it's really interesting that it kind of does that stair step. I'll talk about why. I did a chi-square test, like I mentioned, of independence to see if this pattern, this stair step pattern, was actually significant, and it was. My p-value was significantly less than 0.1. It was less than 0.001. And to kind of show that in a visual way, this is a graph of the residuals. So the residuals measure the strength of the difference between the observed and the expected counts, which is what the chi-squared does. And so the darker colors, the dark blue and the dark red over on Hawaii and Kauai Nihau, indicate a really strong substantial difference between those things, between the observed and the expected counts. So the Hawaii was significantly more, with more individuals in the 25 to 50% bin, whereas Kauai Nihau had significantly less in that. And then that pattern is flipped for the 75 to 100 with not quite as strong of a residual, but still definitely a substantial number there. And then switching over to dorsal fin pigmentation, I'm just going to go through the same two plots, but for dorsal fin, this one there isn't quite as like clear of a pattern. There's kind of a lot going on. There's a lot of interesting things going on that you can see, like in Oahu, it kind of spikes at 50 to 75% on the bottom, and there weren't very many individuals with 75 to 100% dorsal fin pigmentation. So basically most of the individuals I looked at were in that, for all three islands, were in that 0 to 25% really limited pigmentation. And I also ran a chi-squared on this, and it was significant at my alpha of 0.01, but it was a 0.09, so not quite as substantial as the difference that was found with the cape cover pigmentation, which is pretty clear here because there's no color on this graph here. It's all kind of smaller, less substantial differences, and the strength isn't quite as high. So it is significant, but the kind of effect size and the confidence that this is a big difference is not there. So kind of discussing what I found, there's some interesting things. Cape and dorsal fin pigmentation, percent cover are associated with location. So there were significant results for both dorsal fin and cape cover pigmentation. That stair-step pattern by distance is really the interesting thing that I found in this study. So kind of looking at from Hawaii to the furthest Kauai Nihau, so there were kind of a line of differences, if that makes sense. Whereas that wasn't the case with dorsal fin pigmentation. So kind of moving on to what we could do next and what this might mean. Because of the high phylopatry and high site fidelity, that may result in unique pigmentation by location. Like we saw, like there's not a ton of genetic interchange going on, and if pigmentation has a genetic basis, those pigmentation patterns are tied to genetics, then that makes sense. And this result is consistent with other cetaceans. Killer whales, most specifically, have ecotypes with unique pigmentation. It's not obviously the same kind of gray on gray that the rough-toothed dolphins have, but it does exist. Another example I found was Australian humpback dolphins in Australia obviously have similar differences in pigmentation over a smaller area than killer whales. Obviously the Australian kind of coast is a lot bigger than the Hawaiian Islands, but it is showing that pigmentation differences can occur even over a small area. And that distinct populations have distinct pigmentation patterns. And I think that's really a really important thing for future research, is that pigmentation is an important thing to look at, especially because we have all of these photo ID pictures that are being used for population tracking and things like that. We can use that for a lot of other things too. So future research, always a larger sample size. Something I kind of kept thinking about as I was working on this is why, what purpose does unique pigmentation serve? And I don't think we even know that for people, for like why we have different fingerprints. So why is that happening? And more research into this island group. Like I said, there's not been a lot of pigmentation research in this species or in others. So always more in that. And another thing I thought of a lot was differences between this insular island group and the pelagic dolphins that are nearby. Obviously they don't live around the islands, but just kind of in the nearby area. I would guess that there would be a more significant pigmentation difference because those groups are even more distinct and don't intermingle at all. And pigmentation patterns on other parts of the dolphin's body. I really like this picture because it shows that the area that I looked at is a really small area, and there's a ton of unique pigmentation all over these dolphins' bodies, and so there could be even more patterns and differences going on there that it's really hard to see, because when dolphins come up for air, all you really see is a blowhole and then a fin. So it's really hard to get pictures of this. But with more research mechanisms, better cameras, that may be something we can do in the future. And in conclusion, Hawaiian rough-toothed dolphins have pigmentation patterns that are associated with their island of origin, the island where they spend most of their time. And pigmentation pattern differences may be a useful tool to analyze distinct populations and understand genetically distinct populations. I want to thank John Withy. I want to thank Cascadia Research, Evergreen, and the MES program for bringing me back in after a couple of years and helping me to complete this thesis, and my wonderful family.